I'm going to present the end part of the second week of, the end of the second week of Dale Perv's Visual Perception in the Brain course. It's a 4-week course. And this week I'm going to talk about what he has to say about our perception of colored vision. Throughout the course, there's some major themes, one being that what we perceive of the world is not a one-to-one correspondence with what reality is. And in fact, what we perceive of the world is very different from what it really is. He also puts forward his large theory or hypothesis that basically we get around this inverse problem of perception, this fact that we have limited organs to detect reality, and we overcome this limit of information and extrapolate beyond it at a random approach. So we basically, evolution has allowed some of us to perceive inputs a certain way, and those perceptions led to selective success, and then therefore whatever genetic predispositions led us to perceive the world we do at random allowed us to perceive it successfully and live. But it was not such an individualistic learning approach where the brain figures out the world. Okay. Moise, do you have any suggestions for how to load this one?
It looks like an internet problem to me.
Okay. Sorry about this, guys. Okay. Okay. Okay, so on to color. Sorry about everything. Okay, so one remark is that the study of color has garnered a lot of interest over the last couple hundred years. Although color itself, in terms of perception, is actually not that important, given that many animals actually do just fine without it, and they're monochromatic. Or, for example, there's humans who lack certain aspects of colored vision, or color in its entirety, and for the most part, they do fairly well. Of course, color serves function and evolved to do so, but overall, there's been more attention given to it relative to its actual survival importance. So color specifically refers to the perception which is elicited by the spectral distribution of light. And again, note that we're not directly detecting reality in our perception of color. Hue refers to the actual kind of color we see. Saturation is how much of the pigment or whatever makes whatever we're looking at farther from gray. How much color is there actually on a given patch? And then lightness, brightness is just the intensity of that color, right? How light, bright is it? Okay. So a spectrophotometer is how we get an objective measure of the relative intensities of wavelengths. One note that Perfs mentioned is White light is not a pure light. Originally discovered by Newton, pumping light through a prism to find, I don't know, some rainbow effect. And moreover, white lights are not all the same. So daylight and fluorescence and incandescence have different distributions of wavelengths. So in daylight, you have something which has a more equal across the spectra, whereas in fluorescence, you have a greener distribution. Although there's other frequencies in there, there's more green than others. And lastly, in incandescence, you have more red or slower wavelengths. Okay. And so one may ask, so why do we have color, right? Well, color allows us to have another dimension of information to, navigate and act on the world. So, for example, Pervs offers one example of, he offers a colored H stimuli, and he shows you the stimuli in black and white, and everything. There's some random luminance to each patch of the H, and so you don't know it's an H. And then once he adds color to it, you see that it's a red H on a green background. So presumably, color can offer us the ability between red and green, for example, which is the most common kind of color blindness. They don't know that they're looking at an A in this image here because there's no specialty of luminance of these circles creating this A symbol, and so therefore they just perceive the entire thing as a greenish brown, right? Okay. Okay. And so one interesting thing that Dale considers is that, why exactly do we see four distinct hues, right? So we have yellow, we have green, we have red, and we have blue. It doesn't make any sense to say, oh, I have a blue which is kind of red, because that's actually purple and that's a secondary color, right? Now it makes sense to say that you have, a red with some yellow in it, right? Because that's orange, but that's not a distinct hue, right? So the four primary hues, why? And Perf suggests that possibly we have these four distinct hues because it's the only way to distinguish maps of a certain level of complexity. For example, in this map here, if I asked you to color each tile differently, right, and then I could remove the borders and understand each of these as separate percepts, you would need four colors to do so, and then moreover to complete the rest of the puzzle. And this refers to the the four-color map problem, or something like this, where you need at least four different colors to just make sure that no two tiles are the same color touching one another, right? So you need four different colors, so that way you can, for example, not have a yellow touching a yellow or a blue touching a blue, for example. Okay. And so well before we knew exactly what was happening in the retina and what photoreceptors were, Thomas Young proposed that there must be at least three elements in order to account for the light that we do see as color. Specifically, there needs to be like three mechanisms to compare different wavelengths of light, as suggested by Young. Maxwell Field and Hermann von Helmholtz furthered this idea, the trichromacy theory, that there must be three mechanisms within the eye, allowing us to compare the relative wavelengths of light and therefore perceive the different colors we do. So, for example, one reason that led these thinkers to suggest the trichromacy theory, and there must be three receptors, is that you can get any color you want by just mixing different degrees of three wavelengths of light, short, medium, and long. You can get just about any color. So if you manipulate the long wavelengths in some patch, you're going to get something that becomes redder and redder as you add the long wavelengths. And then if you add more medium and medium, it's going to be perceived as more green. And then as you add more, short wavelengths, it's going to become more blue. and of course you have to take away the other light wavelengths. But in any case, you only really need 3, and you can allow people to perceive just about any color. And then, of course, currently there's 3 cone types, which have different sensitivities to the spectral distribution of light, such that you have cones which are most sensitive and fire the most to shorter wavelengths of light, so blues. And then you have cones which respond best to medium wavelengths, greens, and then you have cones which respond best to long wavelengths, or reds and oranges. And then together, these 3 send inputs to the brain and are processed at the level of ganglia to allow us to interpret these wavelengths and make use of colored vision. So as an extension of the trichromacy theory, Hering suggested that perceptually, so not even at the level of biology, but he suggested perceptually what you find is that yellow and blue are actually opponents, and perceptually green and red are opponents, right? And so there's a number of stimuli you can see this with. For example, you may have seen a stimuli like this in the past, where it's inverted colors, and you look at it, and then if you look on a white background, you seem to see the inversion of that, a normal picture. So Dale offers some explanation of this using the center surround idea of a receptive field. So again, a receptive field in regards to a neuron is, what area of perceptual space and what kind of stimuli does that neuron respond best to. And the center surround layout is specifically that there is some positive version of what it responds to in its receptive field, which elicits excitation and firing of the neuron. And then in the surrounding of the receptive field, you get an inhibitory response of the neuron, right? So it doesn't fire as well. So specifically in the context of neurons and ganglia cells in the retina, what you see is a center surround fashion where the ganglion cells, so with their combination of their cones, their respective cones, and you take a region of visual space as it may correspond to the retina, what you have basically is that, for example, in this receptive field where you have, so this particular ganglion cell responds best to green wavelengths hitting its receptors, and then in its periphery, it actually is it inhibits. So this green will inhibit red, and then the red on the outside will inhibit the green on the inside of the receptive field. One explanation for this is just habituation. So for example, this green here, what you get is that as it first sees a green stimuli, so you first hit it with some green wavelengths, it's basically firing a lot, and then it habituates and it stops firing so much. And that's partly due to the fact that it's mostly due to the fact that the photopigments are just died out, and it now needs to regenerate those pigments. But in any case, it's habituating, so it stops responding so much. And in response, the red becomes prominent in the periphery, this red here. But if I send red to the periphery for this ganglion cell, what it's going to do is then inhibit this green cone, right? And so in turn, you get greater perception of red rather than green. And of course, it's not this simple because the brain does a lot of processing of these inputs. But just at the level of the eye itself, there's a lot of processing through these opponents. So for example, in the case of a yellow-blue receptive field for a ganglion cell, what you get is basically an addition of long wavelengths and medium wavelengths, red and green, to produce yellow. So this ganglion cell is going to be, it's going to elicit firing if it is hit by this combination of long and medium wavelengths, yellow. It's hit with this combination, and this causes firing and whatever. And then if you get its corresponding blue cone in the periphery of the receptive field, this is going to cause inhibition on the overall cell. And of course, then this cell, which is kind of sending these, quote, yellow inputs, is going to be inhibited, and then you get an inhibition of yellow overall. Okay, sorry, that was very wordy. but hopefully we can see what exactly is meant by this opponency theory and how are these colors opposites by just looking at this star here for about 30 seconds. So I'll count and just try your best to not saccade your eyes, stare directly at the star. It's very important that all of the cones on your retina are seeing the same patches of light. So do not move your eyes or else this will not work. Okay. And what's happening while you're staring at these, this stimuli is that all of the cones which are taking input from these different colors are basically getting, they're getting very excited at first, and they're firing and they're firing and they're firing, and then their pigments get dyed out, and then in turn, once I move to the next slide, you're going to get an equal distribution of all of these wavelengths, and the cones which are perceiving the opposite will now stand out because the others have habituated. Okay. So keep staring at the star and let me know if this has worked for you. Hopefully what you're seeing now is the opposites of whatever you were seeing previously. So where you were seeing reds, you should be seeing some greens maybe, and where you were seeing blues, you should be seeing some yellows, and where you were seeing yellows, blues, and so on and so on. Of course, one thing to note is that this illusion can be very, very brief, again, because your eyes saccade very frequently without your own control. And so what happens is, the cones were, yes, dyed out and staring at one particular spot, but then once you move your eyes, you're now getting different wavelengths being sent to different areas, and so then the illusion wears out. So it only remains as you're staring at the star. Okay, so contrast and color, color contrast and constancy. So this refers to the fact that you can see spectra which are, for example, the same and perceive them as being distinct, and also you can see two different spectra and perceive them as being the same, right? So they're two sides of the same coin. It's been hypothesized that maybe this is a result of us just having to perceive color in different lighting conditions. And so, what is a red apple has to stay that kind of red in different conditions. And so maybe this allows us to, appropriately navigate the world and whatever. Purves actually suggests that this is not the case. So he suggests that another mechanism for this, an evolutionary mechanism. But okay, so what exactly is meant by contrast, for example? So in this Rubik's Cube here, you may think that this seems a little orangey, like a little brighter than this brown tile, but in fact, they're actually the same level of brown, the same luminance here. It's the exact same. It's an illusion. It seems to be brighter. This is contrast. It's not actually brighter. And we'll look at Purves' explanation of why this may be from an evolutionary approach next. And then, moreover, you can distort this even more by looking at these pattern of tiles here. So we have, so in normal light, you should see these as gray, right? So you may perceive these tiles as being gray under the blue light here, right? So I'm sure everyone sees that these tiles are gray, right? And then in this lighting condition here, these tiles are being perceived as some orangey color, but over here in the blue, they're perceived as red, right? I don't know. If you were to add a red light source, it would be perceived as purple. But what you actually find is that if you then remove this yellow surround, so the yellow surround is actually not being applied here to change any of this, right? So that yellow surround is not being applied. But what you are seeing, for example, with these four squares here, just because your brain is interpreting the yellow surround, it's suggesting that this must be blue. And in this case, it must be this dark gray purple. But once I move the yellow surround, it's actually the same Rubik's Cube as over here. So, sorry, yeah, blue, but not gray, blue, right? Okay. And then here's an example of contrast. So if you're looking at this stimuli here, you should probably perceive this yellow to be a little bit more yellow than this, and this purple seems to stand out, and then this green should stand out relative to that one. And again, if you want to take the center surround approach with the receptive field, you might say, okay, like we have ganglia cell here, which are being very excited by the yellow. So the combination of red and green, sorry, and then in its surround, it's not being inhibited. So the opposite is not being inhibited. You're essentially inhibiting an inhibition, if you will, creating this added contrast that it doesn't really exist. And so if I were to separate these especially, and I were to show you the stimuli individually, you might rate this as being more yellow than this. Now, given the rest of the content explained here, you may perceive these as being the same, but if I were to ask you in separate slides, you might suggest that this is a little bit more purple than that, and that this is a little more green than that, and or whatever you perceive. Okay. So Perv's explanation of contrast and constancy, why is it that we, for example, would see this as being darker than this, even though they're the same luminance? Perv suggests that what has happened is that evolution has allowed us to get around the inverse optics problem, which is that every stimuli hitting the eye is a combination of illumination, reflectance, and transmittance, and each interact with one another such that you can't logically get from the stimuli to any individual term, right? You cannot apply any logic given that you only know what the stimuli is. You only have input. And so to get around this inverse optics problem, Pervs has suggested that at random, some species in our evolutionary history have perceived these things with contrast, have perceived these things with constancy based off of their frequency at which they occur in nature. So what Pervs has done is that he looks at all these little patches of natural images across many, many scenes. And he's done this for many kinds of vision. But in the context of color, he takes a colored image, right? And he takes many samples of natural colored images, right? And then he looks at small pixelation, or small pixels in that image. And then he both looks at, okay, what is the frequency of this kind of patch, a center surround fashion, and how is it perceived? And what he finds is that as you get to a patch of pixels, which are less frequently within these images, so less frequently seen in nature, they become more salient. They develop more contrast, and they're seen as brighter, more luminant, more salient, right? And then conversely, if it's a very common thing, and the most common group of pixels you'll take out at any random patch of pixels aligned with one another, is what you'll find is that the center of the pixels will be about the same in terms of wavelength or luminance or whatever qualities. It'll be about the same as its surroundings, right? And that's because the world itself typically has constancy. And then once you get patches within the world that aren't constant, and you get some deviation from whatever its surroundings are, it then seems to stand out in our perception. And Pervs is suggesting, no, it's not that we are interpreting this thing as being what it is, as just deviating from its surroundings. We're adding to it, right? We're giving it additional salience and novelty because it's special, right? It's not the rest of the background. It's a new thing. It's an important thing, and it becomes salient. So again, as he looks at these patches of pixels and he looks at their frequency, and he says, okay, in a natural image, if you take any random block of pixels, what you're going to find is that the center is going to be more and more like the surroundings usually. But if it's not, what we do is we perceive it as being more salient, more luminant, more colorful, whatever, because evolution has allowed us to make it more salient at random. And this is not us interpreting the stimuli and then logically finding out what its physical properties are, right? That's why we can get all these different illusions. What we're rather doing is we're looking at the rarity of something, and then we're giving it salience, right? Because novelty is important for survival. Okay. That's all I have for this week. So yeah, thank you guys for listening. I hope that